Bismillahirrahmanirrahim. We start the syllabus for the 2022. We have the specimen paper 1 which came in from Cambridge. So I have done the first video on this in which I've discussed question 1 to question 20. Now I'm going to be discussing questions 21 to question 40. This paper's time has been increased, so now it will be for 1 hour 15 minutes. Previously it was only for 1 hour. And starting with question number 21. Bacterial cells divide by a process called binary fission. Please remember this is not mitosis. So one bacteria, the loop of DNA forms a copy of it, and then this divides into two. Well, of course, naturally the two will be a little smaller in size to the original because this is going to divide into two. So this is called binary fission. This is not mitosis. This is not prophase, metaphase, anaphase, telophase. Which macromolecules must be synthesized for binary fission? Naturally, we need the cell membrane, proteins, and the RNA, and we need the DNA and peptidoglycan. But why do we not need cellulose? Because cellulose is not in the cell wall. We have peptidoglycan. Bacterial cell does not have cellulose in it. And enzymes, of course, would not be needed for it because enzymes would cellulose is the wrong one. So that's why this one 3 is wrong. So the answer was 1 and 2 only. Then the electron micrograph shows a group of human chromosomes W, X, and Y. Which label is correct for each of the structures labeled W, X, and Y? Now, if you look at W, X, and Y, the possibilities they've given you is centriole, centromere. Now, you know W is the area where it is held together. So W has to be the centromere. Then if you look at X, these are the ends. And the ends has to be telomere. And Y is, these are sister chromatids. Till they separate, then they are called chromosomes. So the answer was C. Then coming to question number 23, which statement about the behavior of chromosomes during mitosis is correct? They reach the poles of the cell and begin to uncoil. Now you see where you're going to read this question wrong is you did not read this. So now this was wrong. They attached to the spindle fibers to contain them within the nucleus. No, the nuclear membrane has disintegrated. They condense to prevent further translation of genes. No, that is not the case. And they replicate to produce sufficient DNA to form two new nuclei. Two new nuclei. The nucleus is nuclear membrane which is made up. Inside it we have the chromosomes and we have the nucleolus in it as well. So that was wrong also, that it is not two nuclei. So the statement about the behavior of chromosomes during, and the two nuclei would be in telophase, but that means the nuclear membrane, not just about the chromosome. They're only wanting to know about the behavior of the chromosomes. Now coming to question number 24. Gene mutations can occur by substitution, deletion, or insertion. Now this has now been added to the AS level. This was previously in the A2 level. What is the smallest part of a DNA molecule that can be changed by a gene mutation? The smallest part of a DNA molecule that can be changed by a DNA mutation. So you have the DNA and it says ATGC. And by some mistake, this changes. So the whole nucleotide is going to change. The whole nucleotide is going to change, which is the smallest part of a DNA molecule. So DNA molecule is made up of nucleotides. That's the monomer of it. So the entire nucleotide is going to change. It's just not the base that is going to change. The entire nucleotide is going to change. So that is why all these others were wrong. Then question number 25. Which statements about mRNA are correct? mRNA can form base pairs, yes, with the DNA template, with the template strand. Then mRNA can form hydrogen bonds, yes. mRNA is single-stranded, yes. So what we have to understand is both 1, 2, and 3. It was all the 3. It can form base pairs. mRNA will form base pairs with the DNA. If this is the DNA, and then the mRNA forms. So mRNA does form base pairs with it. mRNA can form hydrogen bonds. Yes, with hydrogen bonds is what? with the bases mRNA single-stranded. Then coming to question number 26. The table shows the role of 4 different proteins involved in DNA replication. Protein role: helicase unwinds the parental DNA double helix. Topoisomerase breaks and rejoins the DNA strands. Single-stranded binding protein binds to separated DNA strands to stabilize them. DNA polymerase synthesizes strands of DNA. Which row shows the functions of these proteins? Now they've actually given you that, and then they expect you to really understand if you have really figured it out, which row shows the functions. So helicase unwinds the DNA parental DNA double helix. So let's look at the possible answers. Now, function of this, why did they actually separate? It was to make the strands available as templates. You see, this is the DNA is wound like this. Well, something like this. Now, why would it unwind? It is to unwind because it is available as a template. So that then, of course, now we're talking of DNA replication. We're not talking of protein synthesis. In protein synthesis, the DNA binds to the mRNA. Here is DNA replication, so makes the strands available as templates because they're going to dictate what is here is A, T, T, T. So here would be T, 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 A. Now both of these are going to act as the template on which the further DNA nucleotides are going to attach. So enables tension caused by unwinding to be released, prevents original strands reforming complementary base pairs, adds DNA nucleotides to the 3 end of a growing polynucleotide strand. So it's always in the 5-3 direction. Please understand the DNA replication. It's always in the 5-3 direction. Next. Question 27. Which type of sugar and which type of bond are found in a DNA molecule? So type of sugar is a monosaccharide, and the type of bond found in a DNA molecule is the hydrogen bond between the bases. So monosaccharide, deoxyribose, and the hydrogen bonds. So which type of sugar and which type of bond? You've got to understand that. Then coming on to question number 28, which is a very easy question. The diagram presents transfer section of 3 plant organs: leaf, stem, and root. Now you know 1 is the xylem, 2 is the phloem. Here you know outside is the phloem, and 4 is inside, so this is the xylem. And in the leaf, the upper side is the xylem, leaf upper xylem. That's the mnemonic for it. Leaf upper xylem, and then the lower part is the phloem. So in the root, it says which row is correct for the phloem. In the root, 2 is the phloem. In the stem, 3 is the phloem. And in the leaf, 6 is the phloem. Well, because you see here, you can't see which one is the upper side and which one is the lower side because they're both very... So, but these were the 2 clues were 2 and 3, and this was, of course, then the only answer was. Because here you can't really find, you see, because usually we have a leaf like this, so this is the upper side. So then we know this is the upper side, but here the upper and the lower side were not very clearly demarcated. We couldn't really figure it out because the leaf is very uniform shape. Then we come on to sucrose moves from a phloem sieve tube element into a root cell. Which changes to the water potential and the volume of liquid in the phloem sieve tube elements are correct? You see, sucrose moves from a phloem. So this is the phloem, right? And sucrose moves from into a root hair cell. So this is the root hair cell. And it is moving. Sucrose moves from a phloem sieve tube into a root cell. Into a root, not hair cell, but it's a root cell. So sucrose is moving out. So when the sucrose is moving out, then the free water molecules are going to increase. So the water potential becomes higher. And the volume of water potential becomes higher, but only sucrose has moved out. So the volume of the liquid is going to decrease. Water potential will become higher because sucrose is moving out. So solute is moving out. So water potential is going to higher and the volume of the liquid is going to decrease because all the solutes are moving out. Question 30. Which statements about water movement in plants are correct? Water can pass through cell walls containing layers of cellulose fibers. Water can pass through cell walls containing rings or spiral of lignin. Water cannot pass through a band of suberin in cell walls. So all these three are correct because the apoplast pathway and spirals where there are areas where there are these pits where this water can move out. So this was question number 30. Now coming to question number 31, the photomicrograph is a transverse section of a leaf. Which labeled features are characteristic adaptations of many xerophytes? Now you have to figure out which will be present in every leaf. For xerophyte, you see 4 was wrong, Y is a vascular bundle, and 3 is cells of the spongy mesophyll. They would be present in every leaf, but in a xerophyte, of course, what are the characteristics which are very common to xerophytes is 1, 2, and 5. One is this stomata in grooves, 2 is the trichomes or the hairs, and 5 is the thick cuticle. So the thick cuticle on the surface. So 1, 2, and 5 is the answer, but 4 and 3 would be present in every leaf. They aren't. This is the xerophytic adaptation. It would be present in every leaf. The xerophytes are the ones which survive in desert conditions. So they would have adaptations which help them to conserve water. So there's less transpiration. Then coming to the next 32, a maize crop is growing successfully in a field in which the water potential of the soil is minus 40 kPa. What is the most likely water potential of the cell sap in the root hair cell? Now, when you read such a question, always look at a very important point is what is the most likely water potential of the cell sap in the root hair cell? Now, this was given to you of the soil. Now, what are the choices here? This is more, this is the same, and these are less. Now, you know that the maize crop and the soil water potential has to be what has to be, so why would the water potential of the soil is minus 40 kPa? What is the most likely water potential of the cell sap in the root hair cell? So this is the root hair cell, and in the soil is minus 40. So inside it had to be minus 60 because this is a higher water potential. So it goes from a less negative to a more negative. So the water will move from a higher water potential to a lower water potential. In the case if it was minus 40, then there will be no net movement. This would be the other way around, and minus 20 and 0, it would sort of move out then. So you had to understand why it would be minus 60. Minus 60 because inside the root hair cell would be minus 60, outside minus 40. So minus 40 is less negative. Less negative means it has a higher water potential. Next, coming to question number 33. The contraction of the heart is coordinated through electrical impulses passing through the cardiac muscle. What is the correct order of sequence of these impulses? So you know it has to be sinoatrial node, right and left atria, atrioventricular node, and then the Purkinje tissue. So it has to be here where it went wrong is sinoatrial node, right and left atrium, Purkinje, and then atrio AV node. That was wrong. You see, if you know the heart diagram, this is the SA node. Then this is the AV node. Then we have the bundle of His. And then we have the Purkinje tissue which goes up in the walls of the ventricles. So the sinoatrial node, then the right and left atria, then the Purkinje tissue, sinoatrial, right and left atria, AV node. This is the AV node. This is the SA node in the wall of the right atrium. So this is the SA node. Then coming to question number 34. Which features allow the aorta to withstand high blood pressure? You see, what you have to understand is that the aorta has to be very thick walled, and it has got these crinkled endothelium, and it has got the muscles in it, but it has got the elastic tissue and collagen in the outer tunica externa. So the smooth muscle, the choice between was this A and B. The others were of course all rubbish. This was all wrong, and this endothelium was not going to make any effect on blood pressure. So how does it withstand such a lot of blood pressure is because the wall has to be very thick. Now I would have to be choosing between A and B. You see the collagen fibers and the elastin fibers. You see, elastic means it will stretch and recoil, so it won't burst. The muscle is not going to help it. The muscle only helps in diversion of blood flow. Like for instance, you are exercising on the treadmill, so more blood is needed to your leg muscles. That is when the smooth muscles in the wall of the arteries will contract or relax. So there will be vasodilation and vasoconstriction to divert blood flow. So some parts of your body will vasoconstrict and the leg muscles, the arteries will vasodilate so that more blood is entering the leg muscles. So basically the smooth muscle is only there for diverting the blood flow for one part of the body to the other part of the body so that more blood is flowing in a certain part, like you've just had a meal. So more blood should be going to the stomach and the intestines. So less blood will be going to other parts of the body. So that will be vasoconstriction. You enter this room is very hot. Vasodilation of the skin arteries, skin arterioles, so that more heat is lost. So please understand what they are asking and how you've got to come to the answer to this. Then coming to 35, which row is correct for the pulmonary artery? Now pulmonary artery, as you remember, is the one which is carrying the deoxygenated blood to the lungs. So you're only narrowed down to C or D. Deoxygenated, thickness of the wall, artery, shadow thick, lumen size small. It's an artery. Just because it's carrying deoxygenated blood doesn't mean that the artery will become a vein. This is a misconception. An artery is an artery and a vein is a vein. So the wall has to be thick and the lumen size has to be small. Then coming on to question number 36. The graph shows oxygen dissociation curves of adult hemoglobin in different carbon dioxide concentrations 1 and 2. Which conditions could change the shape of curve 1 to the shape of curve 2? You know this is the Bohr effect. But they didn't ask you what was the name of this. They said what conditions. So it has to be increased carbon dioxide. So you know it has to be either A or B. More the carbon dioxide, more the hemoglobin percentage decreases because hemoglobin gives up its oxygen. Because when the carbon dioxide enters, forms its carbonic acid that dissociates into hydrogen ions and HCO3 negative ions. And the hemoglobin gets attached to the H, so gives up its oxygen. So increased carbon dioxide concentration causing a decrease in pH. Yes, because more carbonic acid is being produced. More dissociation into hydrogen ions and bicarbonate ions. So this was question number 36. Then coming to 37. What maintains the necessary concentration gradients for carbon dioxide and oxygen in the lungs? So basically it is the circulation of blood and the ventilation of the lungs. Why would always carbon dioxide be less and oxygen more inside the lungs? Why? Because we need it to diffuse. The blood is containing more carbon dioxide and must diffuse into the alveolar space. The oxygen must diffuse into the blood. So good ventilation of the lungs is going to ensure that this gradient is maintained. Gradient means this is the alveoli and this is the capillary which is coming around it. Just a diagrammatic view of it. And so if the blood is coming here, it has more carbon dioxide. So there should always be less carbon dioxide here. And there should be more oxygen here. Why? Because this oxygen has to diffuse into it. So basically there are two things which are maintaining this gradient, the circulation of blood and the ventilation of the lungs. Question 38, which cells and tissues are present in the trachea? So there's ciliated epithelium, there's goblet cells, and there's cartilage. All three of these are present in. So the answer had to be B. Then 39, the global mortality figures for some disease in 2013 are shown in the table. How many millions of people died in 2013 from bacterial diseases? Now I'm sorry, HIV/AIDS is a viral disease. TB is only a bacterial disease. Malaria is caused by Plasmodium, which is a protozoa. It is a protozoa. It is not a bacteria. This is a misconception if you think that. Cholera is caused by bacteria. So you added 1.40 and 0.02 and the answer was 1.42. So we're checking whether you knew what are the causative organisms. If you didn't know that, well, you couldn't have done this question. You couldn't have just guessed it. Why is it difficult for B lymphocytes and T lymphocytes to respond to antigens on pathogens? Now, please understand that this is a virus. This is a virus. It is a pathogen. It has antigens on it. Now it says, why is it difficult for B and T to respond to antigens on pathogens that are intracellular parasites? Now, if this virus is inside your body cell, if this virus is inside your body cell, like the plasmodium hides inside the red blood cells. So basically, the lymphocytes do not encounter the antigen because it is covered by your cell membrane. So it is inside a body cell, intracellular parasites, inside the cell. So the lymphocytes, how will they respond to it? Now, all these things were wrong. I mean, when you read the question, it's saying, why is it difficult? They're talking about the B and the T lymphocytes. You're talking about something else. The antigens are constantly mutating. That's rubbish. The antigens can destroy the lymphocytes. But they're saying, why can they not detect it? This is also wrong. Do not recognize the antigen. They are inside it. How they do not even encounter it. So how are they going to recognize it? Because the lymphocytes are outside. They're in the blood. So how are they going to reach this? How this information is going to come to how are they going to get to know that it's inside a body cell? And you've got there actually, that's why that's called antigenic concealment, in which they hide inside. So that completes this specimen paper, and thank you for watching. And please do leave a feedback if there's anything else you would like me to make the videos on. Thank you very much.